Today's lecture is on DNA's structure and function. Before we jump into the actual structure and function of DNA, I want you to recall that DNA falls into that macromolecule group known as the nucleic acids. And remember, these were our information molecules. So these are essential to all forms of life. And if you'll recall, there were two types of nucleic acids that we identified: the DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. And you can see where those abbreviations come from. So here we have a strand of DNA, and notice that it is double-stranded. And then we have our RNA, which is a single strand over here. And again, the function of nucleic acids are to act as genetic material, to store information known as genes, and this is the basic unit of heredity. And cells use these as a blueprint for building proteins. So proteins are the way that cells are able to translate this blueprint into actionable activities or things that need to be built within the cell, jobs that need to be done. These proteins are a sequence of nucleotides, and we'll get into this more when we talk about transcription and translation. But basically, cell has this blueprint, DNA. From that DNA, they make RNA, and that RNA is going to code for specific proteins that control everything about you. All right, and if you recall, all of our macromolecules were composed of they're polymers, or large chained molecules that are made of monomers. Those monomers, or building blocks for nucleic acids, are the nucleotides, and there's one shown here on this slide. A nucleotide at its very base level is a phosphate bonded to a 5-carbon sugar, and then a nitrogenous base. And for those of you that have access to a modeling kit, our modeling kit is using these white tubes as the phosphate group, and these black connectors with that pentagon or 5-sided shape as our 5-sided sugar. And we'll get back to that in a second. But again, a nucleotide bonded together multiple times will form a nucleic acid. And there are 5 different nucleotides possible based on these nitrogenous bases. It's going to be adenine, thymine, cytosine, guanine, and uracil, which we commonly abbreviate just by the capital first letter of those words. So again, the nitrogen bases, this N right here, are going to be the A, T, C, G, and U that you see. We'll get into that more in just a second. So for those of you in class, with access to the modeling kits, you're going to now build 14 separate nucleotides. And using these pieces here should look something like this when you're done. All right. Now, nucleotide chains form these nucleic acids. And remember, this is called a polymer when we have these repeating segments over and over again in a molecule. For DNA, again, as we saw on that first slide, it's a double-sided structure that naturally goes into a double helix, so it's kind of like a spiral staircase. And the rungs of that staircase, or those nitrogenous bases, are going to be adenine, cytosine, guanine, and thymine, or ACGT. For RNA, again, it's single-stranded, so it's like a one-sided staircase. And the nitrogenous bases in this case are going to be adenine, cytosine, guanine, and uracil. Okay, they're really strong bonds between the phosphates and the sugars. Okay, here's our RNA, single strand. All right, for those of you in class, you're going to use your nucleotides to construct a 7-rung ladder of DNA. So you're going to build the left-hand side using those nucleotides you created earlier, and you're going to use this base sequence. So A, C, A, T, C, G, C. And it should look something like this. Now I want you to figure out what the complementary strand would be. And that's going to be TG, TAG, CG. And it should look something like this. Now here we have, again, the DNA and the RNA strand. And I'm showing you what each of those molecules, the cytosine, guanine, adenine, thymine, and uracil, look like when in actuality. So we're abbreviating it with a letter, but this is actually a molecule that's composed of a ring of 4 carbons and 2 nitrogens with some oxygen bonded and nitrogenous or amide groups bonded off of that. Again, you don't need to know all of this. I'm just showing you the detail. We simplify this again for modeling purposes, but these structures are quite complex. All right, the structure of DNA. Okay, again, these are the nitrogen bases that are found in DNA. There are 4 different types of bases: the adenine, guanine, cytosine, and thymine. And the base pairing rules are adenine always binds to thymine, or A goes with T in DNA, and cytosine always binds to guanine, so C always goes with G. You can remember this by remembering that a car goes in a garage and apples go in trees. Okay, so that's a little memory tool to help you remember those base pairs. Chargaff's rule, again for this class, you only need to know this, is that DNA from any species of any organism should have a 1-to-1 stoichiometric ratio of purine and pyrimidine bases. So that just means that if your DNA has 5 million adenines, then you're going to have 5 million thymines. You're not going to have any lone thymines. And the same is true for cytosine and guanine. Okay, and here's that base pairing in action. Notice that there are 3 hydrogen bonds that are going to hold the cytosine to the guanine in a strand of DNA. And then there are 2 hydrogen bonds that are going to form between the adenine and thymine. All right, to get a better idea of the history of the way that we actually were able to discover DNA's structure, you can read through the following slides, or you can watch the video in the next assignment. So here's a picture of Rosalind Franklin and her X-ray diffraction photo of DNA, or photo 51. She took the picture. Watson and Crick deciphered what the picture was actually showing. They discovered that the structure of DNA was a double helix, or a spiral staircase of 2 strands of nucleotides twisting around a central axis. So again, DNA can be described in its shape as a double helix or a twisted staircase. And again, watch that video in the next assignment for more information.